Get started. Sorry for the delay. Apparently the campus is being excavated, so they prevented me from walking across the campus to come to the here, so it will be part of the this seminar. So we're pleased today to have Joohyun Kim, who is a member of the Center for Computation and Technology at LSU. He's an expert on biocomputation and bioinformatics. The last few years, he spent a lot of time on developing tools for high throughput sequencing analysis, and he has applied them as part of projects at Pennington as well as the LSU Ag Center in terms of plant genomes and others. And he has developed certain tools that I think would be readily available to everybody through a website gateway that is developed in an effort, collaborative effort with the CCT as well as the LBRN, the Louisiana Biomedical Research Network, a grant funded by NIH that Dr. Tom Fly is part of this. As part of our own efforts at the vet school, we already have a high throughput sequencer in place and people will be trained on that. Software will be disseminated to all faculty and it will be readily accessible by postdocs and graduate students. And we envision in collaboration with Tulane Primary Center to also add an ion protein high throughput sequencer and additional software to sort of get into the era of bioinformatics. And Joohyun Kim will be instrumental in helping us analyze these data. So without further ado, Joohyun, it's yours.
Yes, good morning. I'm Joohyun Kim from the Center for Computation and Technology at LSU. So I'm going to talk about today DARE-NGS, which is Science Gateway for next generation sequencing data analysis. Especially our unique approach is to provide very viable means for scalable and extensible data analytics. So I'm going to talk about Science Gateway, and I need to explain briefly what Science Gateway means. Generally, it is considered a community-developed set of tools, meaning uniform interface and the standard-based computational the softwares, and sometimes it just means some portal, so you can access with your web browser and then do the many interesting calculations and community recognize important for standard the analysis. So, but I want to emphasize our approach is a bit different from others because our Science Gateway has been developed for considering a bigger picture, cyber infrastructure. So cyber infrastructure is not only talking about software, it actually implicates resource and entire environment. So I also emphasize the last sentence, and it also should consider the roles of human resource. For example, the developers like us, and also scientists like you, and partly like us, should understand scientific goals and technology. So it's not technology-directed oriented approaches. Sometimes there is a huge gap between the developers, software the developers, and the real users, the biological the scientists like you. So I'm going to talk about first kind of introduction on next-generation sequencing platforms and what kind of computational requirements are needed. And later, I'm going to talk about collaborative efforts for our DARE-NGS development in 3 different categories, but mostly focusing on the first 2. And the last part, I'm going to talk about a little bit about my perspective, why NGS data analytics are unique and more challenging than other areas of bioinformatics, for example, for microarray data analysis or some kind of the functional genomics, which has been the main tools for many, many years in this community. So we have a website, so can easily access. I'm going to show brief demo at the end of this presentation. And basically, it's not just simply web page you can easily build. It is an infrastructure, meaning each end user with their own device in the long in some future probably we provide iPhone, iPad, Android interface too. So with your the device, you can access our web page, and then you build your own calculation, and then this our science gateway take your scientific goals, running target application on any machines. Okay, remotely, if you have big machines like our Lonnie system, you can run your calculation there, so you achieve more efficient calculations or deal with a large data set. Or we are actually working on that almost complete test, and we am now the implementing. For example, Amazon or other cloud environment also utilized. So the size of calculation or the size of the data size will be unlimited as you can pay. Okay, so these are the papers and presentations that we made, and some scientific works with collaborative efforts that I'm going to tell is on the way. So these are very intensive team effort, and especially with Shantanu Jha, who was the Cyber Infrastructure Development Group Director at CCT, now the junior faculty in Rutgers Group, Rutgers University, and Na-Young Kim sitting on this room, and other actually the developers. So next generation sequencing platforms are around, as the Gus mentioned. I don't know, you already have that, and the Ion Torrent?
Yes.
Yeah, so that's a new, and I think the development in NGS community. But basically, you can see some the high-end NGS platforms are already available. So especially, I have been working with the Pennington ABI system, solid system, and also some Illumina based the platform too. Okay, LSU has Ion Torrent, I think in biological science, and also you guys have one. So I'm excited to have another opportunity with a new platform. Okay, so this is a brief overview, how actually sequencing data generated, and then what are you expecting to carry out. So depending on your experimental purpose, you might consider genome resequencing, with a new species or new strains, So you want to see whole genome sequence. Or you want to understand transcriptomes, then you need RNA-seq protocol. And also you want to know DNA-protein interactions, for example, for chromatin structure or the transcript factor binding. You actually prepare your sample the satisfying ChIP-Seq protocol. Then the fourth thing is actually you need to carry out computational tasks such as alignment or assembly. Okay, this is the first step you have to carry out regardless of your protocol. And then you have more advanced in data processing for example, genome variation study and or transcriptome profiling, and all kinds of protein DNA interaction and evidence. So which are eventually used for your biological discovery, comparative genomics and disease and cancer and development, the biology, even stem cell and and many other things. Okay, so for those kind of computational tasks, you can see in a huge number of the available resources around the programs. And some are commercial, but mostly open source, so you can easily get, download, install. But as you see, it's not simple task. You need a lot of the programs and broad range of knowledge to choose one. But unfortunately, most people choose one software they know what somebody installed on your desktop. So that's a limitation behind. And also I want to emphasize that's a very risky business because the nature of next-gen sequencing still very challenges in the first step. The meaning, even though you gonna have some kind of sequences from very short read, which generate from NGS, should be considered with a grain of salt, because the computational step for carrying out such a step is not really established well because of short read length. Okay, so anyway, so some people are interested in to provide more integrated environment like Galaxy or some kind of GATK bioscope. Actually, it provide many different tools in one environment, so you a bit feel comfortable, okay, to choose tools and utilize them. And DARE-NGS actually is in the same kind of the perspective, but we actually aim more, I think, in the challenging issues to provide real, complete, comprehensive environment with which you can scale up any size of data and data measurement. Okay, so let me summarize a bit. So why computational requirements are important? Because the next-gen sequence platform produce huge amount of short read, not long read. So you need to work with short read to assemble or map to the known reference genome. So, and also after that, you need a lot of different kinds of process. So it's really compute intensive and data intensive. So it requires immediately, your desktop is not sufficient. Even though you see a lot of tutorials and then small sample data, you can actually use those programs for your data, but you immediately find for your real data, your desktop computing is not really a solution. And this is data-intensive computing. You need more and more data set. I'm going to tell you a bit, but so it's really big data problem. So people are interested in utilizing cloud or emerging the advanced environment. Okay, so for that kind of nature, integrative method is better than just separate individual tools. Okay, so pipeline approach is better than just single tool. So the question is how you build a pipeline, include using many different tools from many different places. But the anyway, solution is you need a pipeline approach. Also, as I just mentioned, you need to have a means to support many different tools for the same task. Okay, so validation of your tools is important. And since this technology grows fast and the significance increasingly important, so you expect the new tools coming quickly, and then you need to build different types of analysis using available tools and maybe some experimental methods or protocols actually requiring different kinds of idea, even though you can find some means to carry out for efficient in a process. You need to update your the knowledge on technology. So meaning it's also challenges. Okay, how you do that, and so our approach is we are going to do that on behalf of you. So you don't need to understand these things and find solutions, everything. Okay, through this infrastructure, you just tell us what you want, and then we build together, and it's going to be used repeatedly and efficiently by your experiment, the scientists, science, and also groups and the community. Okay, so So these are the individual groups that I've been working on for the few years. So you can see there are huge, large scale exome analysis, the data coming from Pennington, and also you can see the microbe. So Dr. Joohyun, plant pathology and crop physiology faculty. So we have long-term collaboration, understand host pathogen interaction, but we did actually the whole genome resequencing and RNA-seq for Burkholderia Glumae pathogen of interest. And also you see Chris Kissender from ULM, we developed a pipeline for ChIP-Seq data analysis. He is interested in having such a pipeline for his ChIP-Seq data, which targets C. elegans transcriptome factor NHR6 binding in the research. Also, we have human genome, especially with RNA-seq for the cancer research. So okay, so this is the first the outcomes with Dr. Ham. So you see the genomes are sequenced, and there is a reference genome from a strain obtained in South Korea. So the first question is how much the strain he got from Louisiana field in the same species different. Okay, so we compared and then see some kind of difference. Especially you look at that the empty space means these are area the Louisiana strain doesn't have compared to the South Korea strain. So huge variations we found. And also there are many downstream analysis, especially I'm interested in protein-protein interactions and protein small molecule interactions. So for example, the red solid line represent the sequence of coding genes. So I took one of them of our interest, which is virus factor related, so-called TOFA transcription factor binding transcript factor. So taking this sequence from my sequence outcome, from the whole genome resequencing, I actually modeled the three-dimensional using the sequence, so we can start to consider structural biology stuff. Okay, so more the kind of brief the summary of the study. So we want to know in a long-term goal rice pathogen interactions. Now we are going to look at rice genome sequencing. Okay, we have microarray data, but we want to go further with genome sequencing approach. But currently we are focusing on pathogen. So we do the resequencing with this. So what we found is there are variations, changes between two geographical strains, and especially we found huge variation in genome structure. And interestingly, they are related to genomic island, which is independently predicted by the genomic island prediction program. So it implicates such variation is actually biologically meaningful and makes sense. Okay, and also we look at individual genes, and then up to now we don't see any big change. So that means even though you have a huge genome structure variation, but the important the coding genes are mostly conserved. So that's a good news. So some part of our observation is not really weird. Okay, so after we identified how they differ in genome structure, so we actually figure out different sequences which is not present in other strain. So this is the experimental confirmation. So you see negative control, so don't see any peaks. don't ask me about the details. I'm not experimentalist, but I understand. So this is positive control. So when you have sequence and this PCR-based approach, so you have the peak. So A means this is a sequence found only from the Louisiana field strain, and then look at those sequence in BGR1, and you see the first sequence can be found in 350GR1, but you don't see that in BGR1, meaning those sequences are missing. Okay, and this is another sequence we found, and you see all sequence we found unique for 336GR1, not found BGR1, implying our the prediction based on the computational process are correct. Okay, so this is in a different way. So sequence from BGR1 as you look at whether that that exists in 3GR1, and you see
Joohyun,
perfect.
may I ask, how big was that genome?
Oh, 7 megabyte.
7 mega.
Yeah, about 7
megabases. And
megabytes.
what was the coverage on that?
Oh yes, I think 100 something. Yeah, it's enough, but it's a bit old. So short read length is short, 2630. So when I do the same kind of calculation with the latest platform, huge difference, and I'm very happy. The depth and the accuracy sensitivity is amazingly good. Right. So comparing to microarray data, recently I tried. You don't know what's going on. Okay. So I did more the investigation. So I look at all coding genes, okay, comparing whether they are conserved. Well, basically this is related to actually the accuracy of your mapping, reconstruction of your genome. As I said, it's not really perfect yet. So that doesn't tell you have some difference in genome sequence of coding gene. It just tells you how you actually compare. But here I just look at non-coding RNAs, and then I found the non-ribo switches actually found in the same way. Okay, so you have 3 and 3 and 1, 1, and so non-coding RNAs, special ribo switch are conserved and found for each. So this actually tells you, the many different type of the analysis you can carry out. Okay, so second example is this is a C. elegans NHR6 binding, the transcript factor. So this is done by Rashi. He's a master's degree, so she came here and working with me and developed pipeline, and she tested and applied the pipeline for his work. So we developed a pipeline, and pipeline is composed of 2 different computational tests. So alignment, as I mentioned, the first rudimentary step, and also ChIP-Seq, the specific peak calling, the calculation. So we make that in a pipeline. So I'm going to show you how it is easily done by you, even though we spent many months for developing. And so she's interested in, for example, so according to ChIP-Seq, the experiment, you can identify certain reason which is candidate for transcript finding with the red line, and this is a genome browser. So this is a location of your genome sequence, but also many genome browser provide the useful information. For example, where is actually the downstream genes are. So you see that meaningfully this location is 5' the end. So this transcription factor probably control the activation of this gene. Okay, and this is interesting observation. So this genome browser provide conserved how much the sequence are conserved among related species, especially 6 nematode. Okay, and then they see huge conservation in this region, and surprisingly, this coincides with this transcription factor binding. So it might tell you this is important part of genome. Actually, you have to keep and across the species. And then genome is huge, and most of them are not really related to what you want. And as you know, the latest development found 98, 97% of human genome is not going to be translated into genes, meaning 98% the entire dark matters are unknown or very difficult to understand. Of course, some of them are in a regulatory system, you may know, but still details are needed to understand. So that's another compelling reason why sequencing approach are the really significant. Okay, so we also do some kind of miscellaneous.
So Joohyun, that chip sequencing was real data, no prediction, right? So basically there was a chip performed,
yep.
Which is basically immunoprecipitation of chromatin.
Correct.
And then those areas that were protected were actually then put
yep.
in the pipeline.
Yep. So Rashi actually is carrying out to optimize protocol for ChIP-Seq the chip actually the experiment. Okay, and then you carry out, you bring those chip data to a sequencing facility, and then they give you the raw data, short read we call, and you do computational tasks like I show, and then finally you got some interest in the outcomes like this. Based on the real data, she found this kind of prediction, meaning so you have many sites actually this transcription factor binds, okay, and that's I think a real beauty. Previously you don't know actually where your transcription factor binds. You just know the reason you already know, right? But this whole genome wide.
Can I have a question here from the web school?
Yeah, it's a question.
Yep.
Yeah. So what happens when you have multiple copies of the same sequence, and how would you determine the distribution for that?
Yes, good question. That's related to the size of your short read and arising from sequencing. If your short read is too small and difficult to understand which part of such similar or repeat unit, still is a big question. So that's why I want to emphasize. If you're working with a big genome, okay, still computational approach is not final. Okay.
So, on chip sequencing, you basically have protected areas of the genome pulled down, and those then are randomly amplified to actually really identify those areas that the transcriptional factor was bound. It was a question?
We're still broadcasting.
The audio.
Audio is not?
We're still broadcasting.
Okay. So, okay, so I'm going to take that question later. That's a very interesting, but it depends on the context you're working on. Some,
Hold on a second. See. Can you listen to us?
You don't listen?
Ramesh?
The problem is there. It's not us.
Hello? You listening?
Yeah.
Can you hear me?
Joohyun, it's not you. It's them.
Can you fix it?
Look, we're transmitting.
Can you fix it?
Don't change it.
Yeah.
I just have to wait for them. They're resetting their audio.
So what about this? Do I have to?
The battery. Let me get this one. But we can still hear you. If the other microphones are taking you off this one.
Can you guys hear us?
We can hear you, Ramesh. Can you hear us?
Yes,
Yes.
now we can.
Okay, we're back. You reset the audio, right?
Yes.
Yeah, okay.
Can you hear me?
Yes.
Yeah.
Okay. So, okay, so that question actually, the answer to that question depends on the context. So some in the cases, you don't worry about that, but some case you should worry about. Okay, but basically, fundamentally, your question is right, and still that's a challenging question because the length of short read still shorter than the size of biologically meaningful size. Okay. So the third thing is RNA-seq, but I'm not talking about much on that. This is a primary focus of current interest. But I'll just tell you, those data are available. So I'm working with that and providing the further the development later. Okay, so I'm talking about what you expect as a challenging problem, what you can solve. Okay, there are many things you already get the solution from many softwares, and this is a very active area with a sequencing data approach, the significance of sequencing data approach. So you can find a solution, but our approach is specifically focusing on some real challenging problem. So I'm talking about scalability. Why you need a scalability? See, big Glumae is just 7 megabase. But if you're talking with human genome, it's going to be 2.8. And also, RNA-seq is the same genome, but data size, okay? So big Glumae, for example, in my data's case, 100 something depth, you get 12 gigabyte data, okay? So you have to understand, so genome size is important for your reference genome, but your data coming from sequencing machines varying depending on your protocol and also size of genome. But human genome, for example, exome is not big, 6 gigabytes, less than your microbe. But generally, you want to consider many, many individuals. So for example, the collaborator work I have involved, they aim to get 100 individuals exome. So you have to multiply 100 times. And if you do RNA-seq, because of different protocols, you need more data from, actually you are getting more data from sequencers, so 23 gigabyte. For example, if you do some more accurate the paired end protocol, you have to multiply by 2, that's the size of your data you get immediately from sequencing facility. Then you have to start with those raw data, right? So data size is big, but also, while you are actually carrying out analysis, it produces also huge amount of data, which depends on the tools you are using. So some people prefer very efficient tool, take short time to calculate, and also generating small amount of data. But you have to be cautious. That is because that's the other way could generate huge data. They want to avoid it. But what if those information they don't generate is important to you? You have to carry out those calculations, which require huge computer resource. Okay, so, and also, a lot of biological discovery has complexity on the different ideas. Combination of your different pipeline tasks. Okay, your goals are different, and also your biological goal would be associated with existing data. For example, 1,000 genome project produce 1,000 individual genome, and it is 200 terabyte. So you cannot download this data even though you want to look at that. So you have to go to Amazon. They are actually generous to donate their resource for this project. Okay, so that indicates in many cases your scientific goals are limited by the ability you carry out, not your scientific the perspective. Okay, so here there's many different tools for only mapping. So mapping is the task the first task you have to carry out. So getting short read, okay, which is bunch of small fragment, and then you have to map those fragments onto reference genome. So eventually you can compare the reference genome and then your the genome, okay? And there are tons of different programs, and then you see different the features. So you are asking why so many different tools? Because I want to remind again, so alignment or mapping is important first step to go further your scientific interest, okay? Then see, depending on tools, you have different results. The numbers are different. If you are expertise to look at each details on the location of, for example, SMP, and look at whether there's a sense or nonsense, then you might come up with less different numbers. But just number from your tools are different, so make you confused. So meaning, you need many tools to validate your procedure. And maybe I'm exaggerating. For your interest, like you don't need many different tools. Probably get much overlap. But this is evidence that I got with big Glumae and also human exome. So there is interesting saying I heard from Galaxy developer. Their team took data from Nature Science and then did again the same kind of calculation with the following same procedure. They all got different results. Okay, that means something you have to be cautious on using tools and then believe your data until you really confirm with your experimental evidence like PCR, real-time PCR, other things. You don't have to believe it.
So Joohyun, I mean, if I understand this correctly, if you're actually sequencing glutamate at the 100x coverage,
yep,
which means you have really more than decent overlaps,
yep,
and the data is really quite strong, why would then these programs have such a great variation in S&P prediction? So the data, the hard data should be identical, right? So there must be errors then.
No, there are many uncertainties. For example, SMP is not necessarily meaning you see sequence one nucleotide variation in specific region, because there are some errors from sequencing and there are some errors for mapping. So you have a tool to predict SMP based on statistical analysis, meaning even though there is a great depth, still uncertainty around, and also you have threshold to assess SMP or not, depending on your program. So what I'm trying to say is the number you just get from the software doesn't necessarily the final conclusion. You have to look at that, and for example, you can compare your SMP to non-SMP database. So you have to verify this is really meaningful. And finally, you have to have experimental verification. This is just in a raw experiment, the computationally processed data. But I suspect with virus, 30, 40,000, yeah, I think you don't worry much on this aspect. This is a eukaryote system and big genome species. Okay.
Hold
so
on a second while I change your batteries. I apologize. Sorry. I have fresh batteries.
Okay. Okay, let's continue. So this is another evidence. In a ChIP-Seq, you have mapping and then you have peak calling based on map reads, because map reads actually are located in transcription factor binding candidate. Okay, and you see that depending on your mapping program, you have different number. Of course, this is number, so you have to look at the location and also FDR or p-value, whether this is meaningful or not. But this tells you, so also you need a lot of different tools carry out, and also validating tools are very difficult. Okay, and okay, I just want to skip. Okay, so let me tell you, so DARE-NGS, the resolve challenges we identified. So we think in a multiple distributed resource could enhance your capability because you can scale your computational size regardless of data size you get. And so our technology based on that kind of so-called high performance distributed computing. And also we build the framework for gateway development. So we're not just developing one gateway. We develop tool middleware with Shantanu, who actually working on SAGA middleware, and we come up with some abstract, the middleware abstract like pilot, job, and the finally there. Okay, and so basically what I want to do is our the infrastructure should be useful for you, okay, not for us. So I want to identify interesting problems, work together, and then disseminate our the development to others. Okay, so I'll show you one example later. So what benefit if you have scalability? You see that if you use workstation, you take 18 hours or 72 hours if your memory is enough. But when I use high performance computing machines, I can scale down to 2 hours. There's a huge difference. You have to go home with this amount of time, but you can get to your lunch. Okay, and so there are some technology we tested for scaling calculations. So I'm going to show the demo. Okay, so how you can use our website. Still not perfect, so I hope you can help to improve that. So if you go to dare.cct.lsu.edu, you can see welcome page, and then you choose DARE-NGS, okay, and count how many clicks actually I make for getting ChIP-Seq data, okay, including everything to access this. So you can see job submission menu. You have several different services. I hope some of them are coming with your collaboration. So host-pathogen is, for example, RNA-seq related program, and BFAST and mapping program. So I'm going to show ChIP-Seq developed with Rashika, Chris. So here, just simple one single form. Okay, so let me tell you. So this is your description. Okay, so I'm gonna put that one, and then if you choose reference genome, so I'm working mostly C. elegans, and then you choose tools you want. So BWA, and this is option for the platform, and this is a peak calling program, and then you choose data set. This is important. You have to send us your raw data, okay, in a FASTQ file format. We can deal with that. Just send your sequencing data, and then we just put the right the sequence. Okay, I just randomly choose. Okay, so one is for treat data, one is for control. Okay. And then also you can choose size of computation, meaning size of computing resource you want to use. So small means it is quick and it's running on the server, but eventually I'm hoping we're gonna present in a cloud-based approach. So if you have an account on Amazon, then you can run your program through here, okay? Even though you pay your credit card. So just submit button and confirm again, and then see immediately your job is prepared and soon running. So you see many jobs previously done. So I'll show you. If job is done, you can see success. Then you can download your output file, okay? Or you can see immediately your output with genome browser that I show, but this is not our software. It is actually located in external somewhere, warm base. So when yesterday I tried, but the server is down. But basically the idea is if you don't have your capacity to carry out some downstream analysis. Somebody provides through web page, we can actually connect it. Okay, so efficiently you make integrative in your own pipeline. Okay, so anyway, so this one I just run and is running on. So you get the status of job. When job is done, you can look at that. Take the download, for example, you can download this, right? And or sometimes it is failed. We can look at it, and then whether this is our problem or your problem, we can actually identify that. Okay, so that's quick demo. Hopefully, you sense how this.
Do you have a demo on the alignment, on the RNA-seq thing?
Alignment. What do you mean alignment?
I mean alignment? The comparison.
Well, this is all actually the numbers. Yeah, numbers. Yeah. But this tells you. So let me tell you once again. So this is a big Glumae in a sequence genome, okay? And so I use 3 different mapping tools, okay? And also you have option to filter your sequencing data if there are some errors so we can apply that or not, okay? And this is the number of SMP predicted by additional tool based on your mapping. And then just numbers are if you use BWA without filtering you got 800 something SMPs. But if you change the mapping program to Novoline, which is commercial program, people believe the best one in many sense.
So all these 3 alignment programs are available through the server? Or
except Novoline.
Except Novoline.
Yeah, we got some the test period, but expired right. one year ago. So because, except Novoline, we have many.
I mean, the Applied Biosystems has their own alignment programs that are on the PGM available on their local server. Are those also loadable on a server format to be run on a high computing environment?
Yes, we can figure out, and I don't see problem, whether you give us the raw short read, so ask us to carry out mapping, or those proprietary mapping program gonna be used, and then give us mapped result, but you wanna do more, then we do with that.
So just wonder the Ion Torrent, which is in this building, because it runs on its own server. Can you do better than what they're doing on their own server in terms of alignments and compare the two?
Well, I don't say the mine is better, but you're gonna justify, you're gonna assess which one is better based on experimental verification. But my approach is I want to provide as many options to you, so you choose which one is better. Okay, so I don't believe one is better than another. It's completely depending on context and your biological goal. But again, developing pipeline is important. That's the one of key parts of our approach. So you come us and then give idea and goals. We develop together. If that's good for the community, then as I show in the ChIP-Seq, this is for Rashika's master thesis. But now everyone gonna use this. Okay, so let me summarize. Okay, let me wrap up. Okay. Okay, so that's summary of NGS, and I show demo. Okay, so this is one single idea that I envision for microbiome system. So you do ChIP-Seq, and then do whole genome transcriptome, ChIP-Seq, whatever, and I don't know, ChIP-Seq is meaningful for prokaryote, but this, you identify the genes and pathway, and then do look at structural biology thing, and then further you can do docking and many binding affinity calculation. So you actually accomplish your own idea, but in a single way, in one stop. Okay, that's one single infrastructure I envision, which grow from the current and existing things. So okay, so thank you for the attention. This part of this work actually supported by an LBRN, just mentioned by Gus, and many collaborators are actually providing their precious data, so I can look at and then see the different type of problems with different data set and the protocol that helps. And also we have some long-term collaborations from New Potential. They are working on iPS cell programming, but they are also interested in some kind of genomic the studies and other functional the genomics for their business. And Dr. Fido Abuela was in biological science, working on non-coding RNA. Actually introduced me on non-coding RNA. And so recently we started a collaboration with Jay Park on computer science. He is going to lead some kind of infrastructure, hardware-wise, cloud environment and some kind of network, connecting, the clusters. Okay, so that's a plan we actually want to build, and then will be led by Jay Park. Okay, thank you. And any questions?
Thank you, Jimmy. I'm sure Dr. Kim will be interested to answer any questions you may have. And
we have one here.
Okay.
Yes, mine's kind of operational with the RNA-seq analysis.
Mm-hmm.
How many replicates would you suggest to get the statistical analysis that you need to call?
Well, I don't know I understand very well on question. This is different from microarray. You need no replicates. Okay, you need one single sample, but your sequencing platform amplify the same fragment a million times, so the depth, the kind of replicate in terms of microarray sense, is your biological same sample, but it's amplified, so it reduces the signal detection, the errors in signal detection. So that's why RNA-seq is shown much better than the microarray cousin, especially comparing ChIP-Seq and ChIP-Seq. So many data shows ChIP-Seq has more accurate sensitivity. Also, good thing is you don't need a priori knowledge on sequence. You just sequence, and then if you find something, that's new. But other technology need a priori knowledge. Unless you know before, you don't know what's going on. So it's based on high throughput and significant depth. So it actually decreases the errors from instrument, not biological implication. But the catch is the caveat is still read length is short, so there are some uncertainties you have to be careful on mapping and some. Yeah.
So what's the size of RNA-seq data, which is genomic data? Because obviously RNA-seq targets pretty much coding RNAs.
No, all transcripts.
All transcripts, but I mean, the transcripts that would be there would be mostly.
But more and more important non-coding parts are becoming very significant.
But the question is, can you do, because the exome genome part is quite smaller.
Smaller, and I think it's very straightforward.
180,000 informative genes, which probably within the capability of even a ion torrent, because you're not going to get 2 gigs or so of database. So you could sequence an exome probably without necessarily going to an Illumina or other system.
Yeah, so here's the number. So exome, you only have 4.G gigabyte from solid system. I show you, RNA-seq from, lymphoma, the cell it was 23 gigabyte, and you need a paired end to improve the mapping procedure. You mean you sequence the both direction. So when you have a short read, you immediately should find the counterpart with the predicted gap. So if you don't find, that means your mapping is wrong. So it improves the mapping quality. So, but it's expensive, of course. So in that case, 23 gigabyte times 2. So compared to this and that, you see, depending on your protocol and the required depth, well, one single solution couldn't tell the entire story. So you have to decide which one is. Ion torrent, I believe this is very efficient if your scale of genome is small or more specified goals.
But the ion torrent won't be able to crank data beyond probably 2 to 4 gigs, so it won't be able to reach the capability of a RNA-seq. Much more. You need an ion proton to actually reach, the coverage that you need for a RNA-seq genomic mapping.
So, the solid system and Illumina GA series or the following one are well mature technology. We know very well. But ion torrent is new, so I'm very glad if you look at together and the capability, limitations, and the best strategy for using that platform. I think that's a great
Yeah, one of the biggest uses that we envision, actually for research purposes, but also for teaching purposes, is really the use of ion torrent for sequencing and resequencing, bacterial and viral genomes. Yeah. So in that sense, you could achieve, 50 to 100x coverage easily. Mm-hmm. Actually, for viruses, you could probably go to 1,000x coverage. Mm-hmm. In which case, your data would be quite, accurate. So we would be interested a lot in, alignment tools and prediction tools relating to, smaller genomes. And Dr. Thun is actually in the audience there. And Ron, I don't know, if you want to share your experience with, your mapping and annotation. Which software are you using in?
Our previous experience?
Yeah.
We went through, the first one was JCVI with, what was it,
Manatee.
Manatee, and what was the other one? The alignment one. And the annotation. There were two different programs we used there to do it.
But you do it through a website from NCBI? Yeah.
At that time, it was Jay Ventnor.
We're currently using Manatee through IGS. It's basically the same base program, but the types of alignment tools that they use and gene calling tools that they use are a little bit different, just computationally. So we're ending up with basically the same results from both companies.
Okay. But those are available through the website, right? They're free?
Yes, they're free through a website. All you have to do is send your data to them, and they'll regurgitate it back to you in an auto-annotated format, and you go in and manually curate your genomes.
Right.
Yeah, one thing I want to emphasize is, so there are many tools like that, and many commercial companies actually provide those services. But let me tell you two, the experience that I have. Of course, this probably not the general. So one is, the Pennington has contract with a commercial company, and they have tons of softwares, and they claim 200 steps carrying out some specific calculation. But generally, they don't keep promise on time because they are busy, and so you can get some useful data, but after 1 month. Okay. Another example is actually, this is also part of our goal. So ChIP-Seq data was available very recently from Pennington. I initially asked them to upload their data to learning machine so I can carry out quickly, but they can't. I don't know, there are many reasons. So we have to wait 1 week to make hard disk. So I went there, pick up one, using my 1 day, and then, Nayeon, actually upload the data from hard disk to our server with 1 day, and eventually the core calculation took, 4 or 5 hours. So, it's not just available software or capability you carry out, but the problem is becoming more complex as your data size increase. So, our actual vision is to prepare the real large side of data, not just providing available tools, so you can easily get.
Okay.
But another thing is we can customize the pipeline with our collaborators. Okay, sometimes, surprisingly, tools used by commercial software company are same to ours. They don't develop all, programs from scratch. They just use available open source. Probably they develop pipelines. Okay, and as the platform, advance, I suspect, maybe 2 years, 3 years, you don't need mapping. Why? Assembly is much better in many ways, if that's working well.
The question is, programs like Novaline, that's commercial, that you guys have access to, are going to be plans to have it available to LSU investigators through DARE? Or are there maybe software packages that could be uniquely accessed? Because that would be very useful if you have those software packages running, on our own, supercomputer environment.
Yeah, I hope so, because to me, Novaline is the truly, single company understand complexity of computation. Their tools are, I think, unique, not only providing the good result, but also they understand the size of computation. Okay, so it's a real situation. If you go to server and ask in a huge calculation, generally, if the resources are not available, they ask you to wait. I suspect any bioinformatics, computational biology related, the software company or small group has huge resources like us. We have huge size of resource available. I just simply use 500 cores, in 2 hours using the machine in Texas supercomputing centers. And cloud is another direction. Okay, if you can pay, the money is not actually high. $20 probably per day for one machine. Okay, you borrow 1,000 machines. You just, I think in a $20-something $100. That's the size is, I think, the biggest size of calculation.
So how do you access DARE? Do you need a registration and password, or is it?
Yeah, so the website I show you still in a kind of the prototype, so you can easily access with registration 1 minute, and then try it. But the problem is you need your own data to understand the calculation is okay. You don't just use the data available, and the privacy is important. With the coming the more collaboration, now you can look at all data set, but you don't know which one is which. But just running single task is not good. So approach me and discuss your interest, and if you find some collaborative effort for your purpose, my purpose, and we can make one and dedicate for your purpose. But I think that's good.
So what's the top commercial software package that does
more and more
sequencing, like Novaline? How expensive is it?
Well, so I think the paradigm is shifted and changing dramatically in this community. Now I believe the top the rank companies, including vendors like ABI and Solid, are interested in integrative approach like us. So they provide not only tools, they provide resource using cloud because they don't have ChIP machines, right? Just contract, and then people pay, and then use the software they develop. So it's not just single software, the standing out among the cloud. now the question is who actually provide integrated approach, and it's not single tool. So if you go to ABI, they trying to keep up with this kind of things. So Bioscope is 2 years ago. Now they call LifeScope. Of course, they charge with this new one, even though Bioscope was free, and something like that. So I think the trend is to provide in a very integrative approach, okay? Not relying upon one single tool. But the question is, in a cost, nobody actually built such a one with free. And also, I think still question is, I believe everyone in this field, well, maybe I'm exaggerating. Everyone in this field don't consider computing resource top priority. But truly, this is a big data problem. You're gonna have a face problem with the size of resource. So you have to be prepared here. Big company easily hire the expertise and build, if they have demand. But I don't know at this point. They believe that's important. So giving you freely.
Hey, Ron, I remember you had some problems with completing gaps in your sequencing. What was the issue there, and how that could be resolved on a
It's really variable depending on the genome. We had was it Photobacterium? We did that genome, and there were 256 contigs that came out of 454 sequencing. Would what we're talking about here alleviate that problem? It's really difficult when there's that many contigs. It's really difficult to close it.
So I suspect the computational requirement task for 454 sequencing data a bit different from the screen data that something somewhat different type of problems.
So if we went to the ion torrent and generated more, would you suggest that we'd have a much smaller problem with contig with the number of contigs?
No, it's not much smaller problem. Different problem. So 454 or the old the sequencing give you long read. Yeah. Okay, so no problem or little problem for confusing location with the lipids or some However, depth should be low, I guess.
Yeah, depth.
And this is a huge depth. So once you have biological implication there, and there is a little chance errors from instrumentation preparation. But the question is,
yeah, what was the coverage on that genome, Ron?
24, I think.
Yeah. So I suspect those depth is not enough for SMP or some very the subtle change in the.
But they had problems on getting the contigs aligned, so they had gaps.
Because it repeats on the end of the contigs, they won't align, or they align too many places.
Yeah, so it could have been some obviously difficult sequence targets, but I think if you had increased coverage, you probably would have eliminated that problem.
Yeah, but cost. That's why NGS actually coming out.
So if you had the NGS and you were going to 50 or 100x, you wouldn't have that problem.
Is there any base pair bias here? Because with 454, our AT-rich sequence actually came back with very few contigs, where the GC-rich was where we had the most problems.
Question again, so...
The base bias, on the sequence platforms. The sequence platforms have different biases. Actually, the torrent apparently has less of a bias than the 454.
Yeah, there are many researches, but because of short read, I think the read error is not significant even though you reach the end of read. Okay. The question is, so you have lower errors from experiment instrument, but you have a kind of uncertainty when you actually map, build onto reference genome. For example, if reference genome really different from your the genome, nobody knows what's going on. So assembly is another. So that's why I mentioned assembly. is significantly important sometime later, as the short wavelength reach probably 100, 200. 100 is currently available, but 200, 300. Then I think you don't need a 454. It's sufficiently long enough solve such confusing location, and with paired end technology and other actually software tools look at candidate, not just map. It's not just search like Google search. They do search initially, but they do carry out statistical inference, and a lot of algorithmic the verification whether you have to keep this read onto the location you found. So not only tools are different in algorithm, but also implementation are different. So it's good to have many, and then you get all data produced by many, and then decide by your biology. I think that's the best strategy. Don't rely upon one single or finite number of tools, because the limitation of the NGS at the current form. So ion torrent, well, honestly, I don't know much about, but it probably provide the way to increase number of different samples quickly. Okay, so you have 1,000 different strains, and then look at comparison, regardless of uncertainties from the current limitation. You're gonna get huge biological significance just comparing that. So ion torrent provided, but sequencer machines, you don't. You have to use all entire lane, so you don't actually combine your sample together. Okay, but ion torrent probably the best in that sense. So different problem again, and tools are available, so you can get whatever you want. But think about long-term goals, so long-term strategy, so you have something better than others.
Okay, Joohyun, I don't know, one more question and then we'll close it. Go ahead, Susan. Go ahead.
I know the Galaxy, they contain quite a lot of programs. Compare this data you developed here to Galaxy.
Absolutely, yeah.
Yeah, they are free. So what's the advantage of DARE compared to the Galaxy programs?
Yes, actually I heard many times for that. Galaxy is popular and more manpower and well-funded, and actually they are computational biologists and the expert on this field. So I respect their approach, but the big difference is they don't consider Galaxy as a cyber infrastructure. They consider this is an integrative user environment. Meaning, once you have huge data set, okay, 100 terabyte, you want to run Galaxy on that, you're going to be a problem. You don't find the resource to run such data set. You can install Galaxy on your desktop, but the size of calculation is limited by your desktop's memory hard disk, not by Galaxy. But that's the point. They don't consider top priority for building their infrastructure. So computer resource, data management are not top consideration for them.
Is it possible to install the Galaxy in our HPC here locally? Then we can increase the capacity.
So who's gonna install them on HPC for you?
You, can you?
Yeah.
Yeah, that will.
That's my answer. Okay. So in reality, so many problems to use Galaxy for sizable big data. They will resolve it.
Even locally.
Okay, with cloud, I believe, because cloud is a big concept. You don't need hardware. I don't know what is the latest in development. When I talk and then heard their presentation, their focus is providing user-friendly interface and many available tools. That's it. So much better than us. But we focus different aspect. We are not competing Galaxy.
Okay, well, you guys can get in touch with Joohyun Kim afterwards. Let's thank him again for giving us a seminar. And Holy shit.
Thank you. So contact me if you go to the website. There is my email. So send me email, any question, and any comment. So I will appreciate it. Thank you.